Hello guys, and welcome to Lab Week. So this is my first video and I hope I will receive a maximum support from you guys. Well, today I'm talking about CLED Agar, which is one of the most common culture media you easily find in the laboratory. We are going to talk about its ingredient, its principle, its appearance, and colony characteristics after culture, and also the type of bacteria that can grow on CLED Agar. I'm pretty sure almost everybody has used CLED Agar because it's a general purpose medium. So CLED Agar is also known as Cystine-Lactose-Electrolyte-Deficient Agar. You can see it from here. C stands for cystine, L stands for lactose, E for electrolyte, and D for deficient agar. Remember, this article is available at www.labweek.com. You can also download this article in PDF. So CLED Agar is a general purpose media, meaning CLED Agar can grow almost all type of microorganisms. You can use it to culture almost all kind of bacteria. It's a general purpose medium. And it's non-selective differential, meaning CLED Agar, though it's a general purpose media, it contains some properties which enables it to differentiate each type of bacteria. And mostly CLED Agar is used for urinary bacteriology, that is urinary culture. It is used for the isolation, enumeration, and even identification of urinary pathogens. And when I'm talking about urinary pathogen, obviously I'm thinking of Escherichia coli, Staphylococcus aureus, okay, Klebsiella, and even Proteus species. So CLED Agar is an ideal medium for urine culture. The medium contains lactose, which is obviously fermented by some of these bacteria, and when fermented there's obviously a color change, right? So this is the basis of its differential. This is the differential property because it contains lactose and an indicator that makes it a differential medium. Also, CLED Agar promotes the growth of all urinary pathogens. The medium also prevents the swarming of Proteus species. You don't want Proteus to swarm all over the culture media. It will make it difficult to identify all the other pathogens. So it contains the electrolytes there's a restriction of the electrolyte which prevent swarming of Proteus species. So CLED Agar also contains L-lysine, L-cystine, which permit the growth of dwarf colony. An example of this dwarf colony is Escherichia coli. As for the ingredient of CLED Agar, we have peptone, we have tryptone, lactose, HM-peptone B, bromothymol blue, which is an indicator. We have L-cystine or L-cystine, which permits the growth of dwarf colonies, and agar, which gives a film membrane to CLED Agar. For the principle of CLED Agar, If let me say this, if you know the composition of a culture media, you can easily determine its principle, because all of the principle is based on its composition. As already mentioned, protein, that is peptone, tryptone, and HM-peptone B, provide vitamins, long chains, amino acid, nitrogenous and carbonaceous compound, with other essential growth nutrients. You know, the idea behind the culture media is to promote the growth of bacteria. We are actually multiplying bacteria. It's just like you are trying to do a PCR, polymerase chain reaction, but here you are trying to multiply a single bacteria. Okay, if you are inoculating, let me say, urine on CLED Agar, you're actually looking for the kind of bacteria or the type of bacteria found in that urine. So CLED Agar, which is a rich media, will promote the growth of this bacteria because it contains almost all nutrients which favor the growth of this bacteria. Peptone is protein. In fact, protein in general is essential for the development of microorganisms. So other vitamins and classes of nutrients are present which actually facilitate the growth of this microorganism. It also contains sugar, that is lactose, which is being fermented by some bacteria called lactose fermenting bacteria, and L-lysine, which support the growth of E. coli or dwarf colonies. As earlier mentioned, bromothymol blue is the indicator and will turn yellow in an acidic pH. So, since CLED Agar is a general purpose media, the type of specimens can almost be all kind of specimen. But CLED Agar is ideal for urinary bacteriology. Therefore, the best specimen to be cultured or to be inoculated on CLED Agar is urine. CLED Agar is in powder form. It has a yellow homogeneous free flowing powder. That's the color is yellow homogeneous and free flowing. And when prepared, you obtain a green colored, clear to slightly opalescent gel form in petri dishes. All these standardized at the pH of 7.10 to 7.50. Guys, it's very important to know the color, the appearance of CLED Agar. I remember back in school, they used to bring us plates of different culture media, and they asked us to identify the media. And it's a hint, because once you know the type of media based on its color, you obviously know the type of sample and the kind of pathogen or the kind of bacteria, the type of bacteria that can grow on the media. For instance, if they gave you CLED Agar, you obviously think of urinary pathogens, urinary bacteria. Okay? So it's very important to know the color of the culture media. It guides you on the type of bacteria or on the type of specimen, because if you were given CLED Agar and you always obviously think of urinary pathogens. Okay, it's clear. There's no way to doubt that. Though it's a general purpose, but once you see CLED Agar, you should be thinking of urinary pathogens. Okay, so let's talk on the characteristics of those bacteria that will grow on CLED Agar. So after preparing your CLED Agar, okay, and inoculating the specimen or the sample, CLED Agar is incubated at 18 to 48 hours at a temperature of 35 to 37 degrees centigrade. So for the type of organisms, you have Enterococcus faecalis, which appears slight yellowish or greenish. We have Escherichia coli, which appear yellow or yellow opaque with deep yellow at the center. Klebsiella pneumonia or Klebsiella species appear yellow to whitish blue. As for Proto species, it appears blue. Salmonella typhi bluish, and Staphylococcus aureus appear deep yellow. Okay? So you should, after culturing, it's essential to always dispose your culture media, right? Because biomedical waste is actually increasing, and nosocomial infection is becoming an emergency. We are having some bacteria which are actually resistant to almost all kind of antibiotics. So it's always essential to properly dispose your media after usage. So if you're interested in buying CLED Agar, you can buy it at from Amazon. You can buy it at $17.17. I think this is cheap from Amazon. Okay. And if you want to download this article, log into www.labwix.com, and you will find this article on culture media. Then you come at the bottom and click on download this article in PDF format. Thank you, and see you next time.